You can actually take the person's blood, make stem cells, and then make those layers of brain structure and put it back to the same person.
Incredible.
And so that is possible.
Yeah.
So there may be even more impossible things we're thinking today could happen. Because actually 10 years ago, it was unthinkable to do single cell RNA-seq. But now we're actually doing that, right? It's almost become a regular technology, routine technology now.
I'm John Foxe, Director of the Monte Institute for Neuroscience here at the University of Rochester. And I'd like to welcome you to another episode of Neuroscience Perspectives. Please like, comment, and follow us wherever you get your podcasts. Today, I'm delighted to be joined by Dr. Xinyu Zhao, a distinguished professor of neuroscience at the University of Wisconsin-Madison and a leading investigator at the Waisman Center. Dr. Zhao is internationally recognized for her pioneering research on how molecular mechanisms regulate neural stem cells and brain development, and how disruptions in these processes contribute to neurodevelopmental disorders such as autism, Fragile X syndrome, and Rett syndrome. We'll talk a lot about that. Her work has earned her numerous honors, including the Willis Distinguished Achievement Professorship, the Jenny and Kyle Professorship in Novel Neurodevelopmental Diseases, and the National Fragile X Research Award. Xinyu, thank you so much for joining us here today on this crisp autumnal day in Rochester. It's such a pleasure to have you on the podcast.
Thank you for the invitation. And it's also my honor to be the keynote speaker for the symposium.
We're so excited for your talk later today. We're really putting you through your paces. We have you here in the early morning, Rochester, doing a podcast, and then you've got to get ready to give a big keynote address.
So I'm honored and excited to talk about research with you.
I'm gonna dive right in here. This is a buzzword in our field now, organoids, and so help our listeners understand what organoids are, how we make them, and why they've become so important to us in the field.
So organoids, actually the word was actually ignored for many years, but only become really, really popular since about 2013, 2014. So when we talk about organoids, we're talking about this ball of cells that we can create from a human pluripotent stem cells. So when you grow them in the culture and give them the right condition, they can actually start going down to a differentiation path. And if you put them together, they can form this kind of spheres, tiny little balls, and they can grow and become different cell types that you instruct them to be. Like, so there are organoids for brain, and we call the brain organoids. There's also kidney organoids, blood organoids, and liver organoids. They're all different kinds of tissue organoids. So what they represent is this kind of mimicking the early developmental path of the tissue genesis. And because they're human origins, so you can actually use them to study the mechanism of development in humans. And you can also study the genes or the protein or the factors affecting development or in disease. So that's why actually they provide this really good model for us to study human development disease. And that's why it's such a exciting kind of a experimental platform for us to use. And what's actually, in addition to that, for many years, we're relying on animal models or post-mortem brain tissue or other tissue to study disease. However, there's a really scarce availability for the tissue. And also, more and more research have shown that when you use animal models, not all the disease mechanism can be replicated in animal models.
So it's similar as their genetics are to us.
And the pathways and the signaling mechanism. Humans are kind of, we evolved into this complex regulatory mechanism that not all of them can be replicated in animal models. So that's why everybody's so excited to have this human model finally we can use to study human disease mechanisms.
So where we came from would have been a much simpler preparation, right? You have a tissue culture, so you grow from stem cells, maybe a specific cell type, a neuron or glia, and it would be flat on a plate.
Yeah.
But this is different. These are spheres. They're complex assemblies of different cell types that actually mimic the tissue in the human
Yes.
brain or in whichever organ you happen to be studying.
Yes, I know more about brain organoids. That's what I use and what I study. So basically, when you grow them, give us some of the chemical patterning chemicals to actually to push them into the brain specific pathway. Then they basically follow that, and they follow this intrinsic programs to develop into the almost like embryonic brain tissue, like 3D structure. So they will follow the path of becoming neuron, generate neuron first, and then astrocytes and oligodendrocyte. And then they also have some additional other cell types. They going through this neurogenesis process that you really can use that to study human development, which we don't have access to, in the early embryonic stage. Yes. So that's why it's a very valuable tool for us to understand human.
I mean, it's completely remarkable. I mean, I think first time I heard about organoids, I was completely flabbergasted. The idea that you could have a three-dimensional structure with many of the important cell types assembled into a functioning unit, right? Because you'll actually get brain activity in these cells, right? If you put an electrode into one of the cells, it's firing away and it's doing stuff.
Yes, so you can actually grow them for weeks and months, and recently there are publications showing that they can grow for 5 years. So then at some point, normally it's after about 4 or 5 weeks of growing, the neurons start to have a neuronal shape and also start having spontaneous activities. Yeah. You can detect that by putting electrode in there, or you put them onto this flat surface with electrode and called a multi-electrode plate, and you can measure the activities. Yeah. And those activities become increasingly more and more intense, more regular, and especially when the glial cells start generating and to support neurons, and you can see even more activities. Yes.
So they start behaving like neurons that are actually inside the brain, and that's incredibly powerful tool then to observe the developmental path of these cells.
Exactly.
And then, of course, they give us a target to shoot at. So how do we use then those organoids to tackle disease? And tell us a little bit about that. What does an organoid do? What work does it do for you in your lab?
So we use the organoid to actually model Fragile X syndrome, and also some of the other genes important for, known to be important for autism, other developmental disorders. Right. So we could actually use patient-derived iPS cells. This is induced pluripotent stem cells from the skin or blood from for example, Fragile X patients. Yeah. We take those cells and using the pluripotent factors, so-called Yamanaka factors, and also other methods, to make them into almost a human embryonic stem cell like cells, and then we make them into this organoids, and we grow them in the lab. And then at a certain time development, for example, what we do is we take them at 8 weeks time period, which is a neurogenesis period, a lot of cell proliferation and the cell genesis. The neurons are all very immature at that stage. We also take some organoids at 20 weeks differentiation. That's when they become a more mature neuron, but it's still, corresponding to embryonic state. But they are also start having glial cells, like astrocytes. And we also take them at 32 weeks, which actually corresponding to around that time is corresponding to birth, because it's human stem cells follow the human developmental time point to develop. Yeah. It's like a human gestation, 40 weeks. So that 32 weeks, they're very close to the birth time of a human. Yeah. And those neurons more mature, we see also a lot of astrocytes and also some oligodendrocytes, the early oligodendrocytes. So what we want to see is in the context of Fragile X syndrome, do we see neurons are not developed very well? Do we see more neuron or fewer neuron? Do we see gliogenesis are affected in the Fragile X syndrome? Yeah. And we also want to see that if there's a synaptogenesis affected, which we have seen in patient postmortem brain, there's
Right.
like a dendritic spine abnormality. Can we model that in the organoids?
In the organoids, yeah.
And because we also take them to we take them, section them, looking at all the neuron glial cells in the organoids. We also take them to do gene expression analysis, and we dissociate them to do single cell sequencing. We also take them to do proteomics and metabolomics
Right.
to figure out what might be wrong, and then we identify potential treatment we can actually apply to the organoids. We treat them with chemicals, or we restore some of the important genes
Right.
missing, or we knock down the genes that shouldn't be there, and to see if we can correct those deficits. So that's actually provided really a kind of a human model for us to
It's an
to
extraordinary.
figure out.
It's an extraordinary tool. Yeah.
Yes.
I mean that part. Just quickly again for our listeners, many will know that Fragile X is it's a well-known neurodevelopmental disorder with a pretty devastating clinical track. So
Yes.
to be able to intervene early would be a huge bonus to medical science.
Yes.
And this idea then that you can actually watch the normal or pseudo-normal
Yeah.
developmental path of these neurons and all the support cells, the neuroimmune cells like the glia and oligodendrocytes, as they develop in partnership, and actually watch the disease process unfold potentially.
Yes, and then the other really important thing about the organoids, because these are derived from a real human patients.
Yeah.
Many of those patients have been undergone extensive clinical assessment. So we have iPS cells derived from patients with stratified by their brain waves and measurement and their behavior assessment, their clinical symptoms.
Yeah.
So we also know how they respond to clinical trial drugs. And so when we generate those human iPS cells, neurons or organoids, we can figure out why some patients actually responding certain
well and others don't.
Yeah, others
it's
don't.
Incredible, yeah.
And so can we, so we are trying to figure out the molecular mechanism under that, and then also try to figure out how we can use different drugs or different combination drugs to treat the organoids.
Yeah.
So the goal is maybe in the future we can figure out that we can stratify patients based on their clinical symptom, their brainwave signature, and then to figure out how to do a combination or targeted treatment, so to improve the clinical trial success.
And this is really, the ultimate in personalized medicine. It would be Jimmy's own disease in a dish, we used to say, but actually in this little living organoids of brain tissue, where you can be designing interventions very specifically for that individual. Is this what works for Jimmy? And then obviously the other piece is that these organoids, you've been working with Fragile X and Rett syndrome, but this can be done for any disease, right? I mean, it's a simple skin punch or a little bit of blood that you can get the iPSCs from.
Yeah, so currently actually derivation of iPSCs has been really kind of become a mature technology.
Yeah.
So our core facility and across country, there are many universities or institutions have their core facilities can derive iPS cells from patient blood, patient's skin biopsy, or some even from a tooth kind of for the children's
we did that in my own
tooth.
lab. We made it made from dental pulp. It's teeming with stem cells. Yeah.
Yes, and then so these iPS cells provide really the tool for us to move forward to study the disease mechanism. So so this is if we talk about the personalized medicine, could be the future. Yeah.
It's completely extraordinary. It makes the hair stand up on my neck. Even though I've known about organoids for a long time, it still astounds me that this is a tool that we have in the tool chest these days. You also you have a leadership role. Actually, I will in a very important network here in the United States. There's for our listeners, there are 15 national IDDRCs. It's a mouthful. Intellectual Developmental Disability Research Centers. Mm-hmm. So there are 15 national centers, and you're at one of the really famous ones in Wisconsin, Madison at Waisman. And then you take a leadership role to run the network around organoids right across the IDDRCs. Would you talk to us about what the importance of a network like that is, and what it brings to the table in terms of our abilities to to get out to rare disease populations.
So that the IDDRC was started by actually the paper signed by JFK.
That's
that
right.
was over 60 years, and this network
because his Irish mammy told him to do it. Rose Kennedy.
Yeah. And so this 15 center network is really important. It's spread out the entire country from East Coast, West Coast, and the Midwest. And the Waisman Center at University of Wisconsin is one of the first center funded by this. We've been continuously funded by IDDRC for all these years. And so the IDDRC bringing together people who study developmental disability and from all different aspects, from molecular mechanism to brain imaging to behavior. And really what our focus is trying to help people with the developmental disability, not just the children, but also the grown, the adult people who actually suffer, because many of the developmental disorders and actually affect they are not lethal, so the people suffer from that. Yeah. They can grow old, and they actually have a more severe deficit sometimes, and especially when they become alone, their parents are all gone. So this network really is very important for us to move forward with research, treatment, and all the advocacy and education of the population. Right. I'm fortunate to be part of the IDDRC at the Waisman Center. And we, about 6-7 years ago, when human stem cell become really popular across IDDRC labs, and we start forming this cross IDDRC stem cell work group, which is led by Anita Bhattacharya, Chris Grohl, and I become part of that. Together, we organize this monthly kind of a Zoom meeting to talk about how to actually effectively use iPS cells. Right. Because Wisconsin's is the university first isolated human embryonic stem cells. And we had a lot of experience in working with stem cells and iPS cells. There's a large group of scientists in there. And our core started making iPS cells actually for over 10 years already, and also provide a lot of information for other centers to make how to make iPS cells.
And that's the key, right? It's that there's expertise that come up in various domains, and obviously Waisman was a world leader in iPSC and stem cell development. And then that you can immediately transfer that information around all the network and get it out to the four corners of the country.
So this across the IDDRC network is really important in terms of that kind of collaborative environment. So the monthly meeting organized by Anita and really kind of we talk about all the problem we may have in the lab. So why my iPS cells not differentiated very well? How to make sure the iPS cells are really at high quality? So because of everyone have sometimes similar kind of questions, or actually one person have a question, another one have the expertise. We really helped each other. And also during that time, and we realized that there's a lot of variability among the quality of iPS cells and how you culture them, how you maintain them, how you differentiate them. So Anita and Mustafa Sahin, which is a director now at Boston Children IDDRC, they put together the first paper across IDDRC kind of article talk about how to effectively, reproducibly use the iPS cells. And also, soon after that, we realized that this organoids become the new kind of really exciting model, and everybody start trying it in their lab. And we realized that this is a very tricky kind of a system. Because depending how the quality of your iPS cells, and also depending how you grow them, how you analyze them, it can be extensive variabilities, and there are limitations also in terms of how organoids can model disease. And so we all facing the similar kind of challenges. Right. So that's when I started to lead this work group, also part of the stem cell group, and we start working on how do we actually effectively use organoids? I also at the time serving on NIH study session, we review a lot of iPSL organoid grants, realized that actually people are having a lot of troubles with organoids, because there's no standardized method for analyzing them. And if you look at the most paper, just dissociate them to single cell RNA-seq. But if you want section the organoids, you can see they're all very different from within.
Right.
Even from the first batch of organoids, one organoid is very different from another one. So then how do you actually quantitatively analyze them?
Right.
And also the growth conditions of organoids. So should we use the media with a less, lower concentration of glucose? Or should we use
right. So it matters what they're living in and the food that they're eating, essentially,
yes.
right?
So we start talking. So we need to put together like a kind of standardized method, guidelines.
It's so important. I don't think this can be emphasized enough. I think maybe sometimes people think, okay, Boston Children's at Harvard and Wisconsin, Madison, University of Rochester, we all sort of compete, we do our things and that. But actually this part, the key here is for the American taxpayer, they get great bang for the buck through these networks because we share this information. We're all on the same path. We're all trying to come up with a standard, a gold standard, and save each other time and wasted effort, right? You don't want 15 different groups, making the same mistakes all the time.
So actually, I have never seen any cross-country groups scientists work so effectively together. So when we got on the monthly Zoom, so we will have like a student will present some of the data and talk about the challenges they have. Like for example, Jason Stein and the UNC, they start testing some of the the method for differentiation. They will bring out what challenge they face. We chip in our experience and we start testing some of the same method they use. So we really have a lot of information exchange.
What an opportunity for that student to have experts from 15 different national centers weighing in on the project. It's completely outstanding.
And then as a part of that, every time, because IDDRC has a yearly director meeting, we kind of piggyback on the director meeting. We always had our stem cell group like sub meeting. We'll have like talks and we discuss what challenges we have. We have a data presentation. We also have technology kind of exchange.
Right.
And for people who cannot attend, we have the Zoom option, so they can Zoom in to participate to also gain the information. So
fantastic.
So we start calling ourselves the Cross IDDRC Stem Cell Consortium, and because we really feel like we are a consortium of people who work together trying to push forward. So then last year we finally finished the organoid review article we publishing stem cell reports, which is a society journal for the international stem cell.
And it's such a great resource. That paper, so I know it's already had big impact on folks. So it's really, really amazing.
So the paper is not supposed to be telling you what to do, but it's really kind of recommendation. We actually have a table recommendation. One of the key recommendation is no matter what you do, you need to give a really detailed kind of description what you're doing. So for example, in the past, when people publish an organoid paper, they will just show the data, but you have no idea how many lines they use, how many replicates, how many batch of differentiation they use.
So this
we
is
recommend,
very important
this
for rigor and reproducibility.
Exactly. So because organoids, even though it's really fantastic, it does have a lot of limitation. Remember, it's very small. It's only up to 4 millimeter diameter, which is much smaller than the human brain. And also because there's no other cell types, there's no blood vessels, there's no microglia. So you actually, those cells, those ball of cells, only can develop into a certain extent of representing human brain.
Right. It's not a perfect representation.
So, and they're variable in many ways. So that's why we have to take that into consideration when we use that model for modeling human development and disease.
Let's turn to some of your own work. you work on environmental factors and how they interact with genetic programs to influence neurodevelopment. Tell us a little bit about that and what you see as the major findings in the field there.
So I always been interested in the environmental factor, for example, epigenetic factors.
Right.
So we have done some work in for example, running, physical activity,
Right.
how that affect the brain development and development disorders. So in the context of when you apply environmental factors, you have to remember you have to control many of the aspects. So we decided to use this model, which is actually the running model, which the mice like to run, and you put the mice in running well in the cage, they will run. They run 10 to 20,000 rotations per night.
Amazing.
And one of the major impact of that running is actually in the hippocampal region. They actually promote this new neuron generation, maturation. They also actually, these mice they look healthier. They actually learn better. So we use this model to study actually what genes are important for driving neuron maturation inside the brain, especially in the hippocampal region, which promote learning and memory. So we actually label those neurons, these new neurons with a tag that can tag the polyribosome, can allow us to pull out the RNA to look at the gene expression. And we identify the...
So just backing up, I mean, this physical exercise intervention changes how many new neurons are being generated in the hippocampus, the memory banks or memory system, and ultimately changes how genes are being expressed. And
yes, so this... if you actually look at the new neurons in the brain, in the running mice versus non-running mice, there's the neurons in the running mice, during the development, they develop faster and they become mature faster. They integrate into the circuit much better. Right? Yeah. So this provides us a way to figure out what genes are underlying that process. So how do we actually identify those important factors driving neural maturation, integration, and which is underlying at least learning memory in the mouse models. Right. So
running makes smarter mice. That's the other.
Yes, exactly. So actually, I remember it was during my postdoc, and my lab mate found that running really trigger more new neurons in the mouse, and that should make mice smarter. And I still remember that when she found that, and then we started running in La Jolla and start exercising. Said, okay, running makes us smarter. Let's do it. Yeah. So and so in this study, we have identified the genes as really important for neural maturation. And so when we pick one of those genes and when we overexpress it in the new neuron, they also make a neuron more bigger and more mature faster. And then when we knock down those genes in the mouse model, and the running mice no longer have the increased neural maturation. So we know that at least that gene is one of the key genes. Right. So in addition to that, we also look at what genes are really important for this maturation of a new neuron during running. And many of the genes are related to actually autism, schizophrenia, and Alzheimer's disease. So it's really kind of unveiled as how environmental impact can really driving gene expression to affect behavior. Yeah. So that has been one of my always something I wanted to do to see if we actually can identify those driving factors for neural maturation, so we can actually help with many of the disease models are actually affecting neural maturation, especially Fragile X syndrome, autism. So
how interesting! That's fantastic. Yeah. Quick message to our public, right? Get out and exercise. Get on your bike, do your running.
I'm trying.
Right? Yeah. Let's talk a bit about you and your journey. I mean, obviously, you're like myself, didn't hail originally from these shores. Is that fair to say? So in terms of coming to America and entering the science enterprise here, and sort of just the meeting a new culture and that, how was that for you? And how do you think about the current, a big question, the current sort of scientific gestalt in the country?
So I was grew up in Beijing, and so I wanted to be a scientist since I was, I think, 5 years old.
Wow.
Because somehow, but my dad is a rocket scientist working on the rocket.
Well,
And
I'm going to interrupt you. Tell our listeners what your name Xinyu means.
My name is Xinyu, and that's my first name. And Xin means new, Yu means universe, and new universe. And my dad, he was a rocket scientist, and so he actually named me after his passion to build a new universe.
Beautiful.
Actually, my brother's name is Build a Universe, and my youngest brother, I have two younger brothers, my youngest brother's name is Travel in the Universe. So basically, all of us have something to do with universe. Yes. So
how amazing! It's just such a lovely story.
So I somehow remember in my vague, very early memory seeing my dad in white lab coat. But I don't know how that happened because his work is behind, like a guard the door.
Yeah.
I never was able to actually go into his work, but I remember seeing him in white lab coat, maybe a photo. So I wanted to be a scientist and want to wear white lab coat. That's my dream for all those years.
Wow.
And I went to Peking University and I majored in biophysics because I like biology and also physics. And during the time, I became really interested in genetics and molecular signaling. Mm-hmm. So I came to the U.S. and entered a PhD program at University of Washington in pharmacology.
Right.
And I loved just doing experiments. I feel like this is just the world of science open to me because at the time, U.S. is still now is leading the science or scientific research, and there wasn't anything available in China then, and so I loved just being able to do science and focus on doing that. I remember working late hours. I remember running HPLC overnight, just try to get all the, like every hour doing injection and collect samples and overnight and then try to get an experiment done.
You'd found your lane, right?
Yes. So I just love that. And I also see that in some of the younger generation scientists and the new students. So, but the kind of a little sad thing these days is that we actually, with the current atmosphere, there are fewer and fewer those, foreign scientists are really willing to come to U.S. And I feel like we're depriving really kind of
the lifeblood of the U.S. science engine.
Yes, and
to a degree, no question.
Yeah. So, and also I have, over the years, I have trained scientists. Some of them stayed in the U.S. Some actually went back to China as a faculty there. And so now we cannot collaborate with them. And in the past, I collaborated with my past trainees and with publications and the student exchange, information exchange. Now we cannot do that. And it's also really sad because science is really global. Especially when you work with rare mutations, rare disease, you do need to have all the information across the globe to actually to generate that information.
And hard to see, and I don't want to get political or anything, but hard to see how trying to cure devastating neurodevelopmental disorders has any role in security or anything like that. I mean, this should be everybody on the one boat rowing in the same direction, really.
Exactly. Right? And especially, for example, my former director, Albie Madison, is working on Alexander disease. It's a very, very rare disease. And he created a consortium across the globe, and the people get on Facebook and talk about, what their needs. And they now, so he actually identified GFAP was the gene, and now they actually have a clinical trial on the treatment now. Right. So, but without this cross globe and the
back to our networks and the importance of this, that it can't. Very little gets solved at the level of a single lab or a single university. It's the global science community.
Yes.
Where the US has traditionally had the main leadership role. We can't let this go, right?
Exactly. I think by closing the door, basically you're kind of isolate yourself, and the isolation has never been good for any country. And being a Chinese, I know that because over 100 years ago, that's when China tried to close our door. And the fact is when we become backwards in technology and we get bullied, right? So any country who close their door will be passed because the world is moving forward. Without you, they're moving forward. So I hope that in the U.S. scientific community and the government would realize how important we have been as a global scientist, and so we still can lead that rather than being put behind.
Yeah, well, I appreciate you saying those things because it's an uncomfortable space to speak about these days. Tell me, so you arrive in Seattle, Washington, right, for graduate school. Had you been in the U.S. before that?
No.
So you come off the airplane.
Yes.
You're in your 20s at this point?
I was in this country when I was 22.
22.
Yeah, so...
How was that? What did that feel like? Yeah.
So I was very excited.
Yeah.
So I feel like suddenly I'm on my own.
Yeah.
And I can do whatever I wanted.
Yeah.
And so I was in the graduate program. And also I feel like, now this is the world that with very strong individualism.
Yeah.
And no one tell me what I could do, what I needed to do. I just need to do my good science, which I love the science part. I love the research. And also I started exploring other things. Actually, I did a lot of mountaineering, biking and sailing.
Excellent for your neurogenesis.
In Seattle. I climbed Mount Rainier when I was there, and I was part of the Boeing Alpine Club and joined there and learn how to actually climb mountains with, ice axe and ropes and stuff. Yeah, so I really enjoyed being a grad student.
And tell us, how was your English when you arrived?
Oh, that was so I went to probably the best schools in China, and so I thought my English was good.
Yeah.
Like I had a lot of grammar classes.
Yeah.
But I only in university we had one professor who's actually a native English speaker. Before then, it's always a Chinese professor, Chinese teachers, and so I never really had a chance to talk to anybody from a English-speaking country.
Yeah.
So I remember the first day, went to the department, and I practiced to say that I'm Xinyu Zhao. I'm a new grad student, but I couldn't really say it. I just stuck to my throat. I couldn't say it. No. So the person, I remember the administrator has to actually, you're the new grad student. So yeah, so that was not easy. And also the first year classes frequently, I remember I actually had a little tape recorder. I would record it and listen again and took notes later.
Because it was going very quickly. Yeah, what a challenge.
But the nice thing is I had a very strong science background from graduating from Peking University. We had really hard classes.
Yeah.
So all those things came back to me once actually the class wasn't hard. It's more the English. Actually listen to the lectures and taking the notes. And because of the recorder, it really helped me. And I got all A's for my classes. And so I remember my biochemistry teacher in the beginning, and she wasn't very sure about me. And then when I got to the top score, and then she started treating me differently. So, that was not easy the beginning, but quickly I think and I learned. And also, watching TV helped me too. And I watched Cheers when I was. That's every night after study, before going to bed, I watch Cheers.
Yeah.
So I learned a lot of English from Cheers. Sometimes I use those English inappropriately. So because they said, you don't say that. That's a bar language. So last year when I went to Boston to ICR, that was the first time I went to Boston, actually. So I went to Cheers bar, and I said, wow, this is a place actually I watched on TV for all those years. So yeah.
So your English is from Shelley Long and Sam Malone and Norm Peterson Clippy.
Yeah. So
you know, that's fantastic. What a great show. It's personal to me too, you know, as somebody who arrived at 20 years of age with this wide open possibility. I mean, there's a lot to be said for what the US represents for people who come to study and to do science.
Yeah.
And as we were implying, may it last forever, because boy, has it been the platform upon which, you know, so much has been done. I mean, in the time you've been here, what we've witnessed from the US science engine. And of course, it's the best part in some ways is it's translated back into our home countries. People came and trained and went back, and that's a good thing. You know, we didn't lose anything in the US by that. We gained by having collaborators and scientists around the globe all working on the same issues.
Yeah, exactly. So I would love to be able to collaborate with my Chinese colleagues.
Yeah.
So, and I hope that still can happen sometime.
Those days will come again, I'm sure. Yeah. Do you feel hopeful now, or are you worried with where we're at in science?
I am, in the sense, you know, because I have children. I watch that, you know, because my brother has kids in China, and so I feel that science education has always been, like in the grade school education, is not strong enough in this country.
Yeah.
There's not in middle school, high school, the most popular people in the schools are athletes, not the people who actually win math contests or actually science Olympia contest. So I feel like the science education need to be stronger, but U.S. has been always benefited from all the scientists coming from foreign country, educated in Europe, in China, Japan, Korea, in India. You know, we all come here because U.S. provide this fantastic platform for us to explore, to really work hard, to establish, to learn.
Right.
And if we start stopping that, and yet we still have not really boosted our own scientific education, then how are we going to be leader or stay as a leader in science? So that's actually, I think, it's worrisome in that. I feel like the policy of the government can make a big difference in this.
Of course.
You know, many countries are really put education and science as their top priority. And I feel like this has not been...
We're going the wrong direction on that front.
Top priority here. Yeah. So,
where do you see stem cell technology now in 5 years? Are there things looming on the horizon that are going to blow our minds? Or
actually, the stem cell technology, I think, for example, we talk about the organoids, the limitation, but many people are working on making it better. Model. Like, for example, like this morning, Guoli Ming is talking about the technology that her lab is developing to make organoids grow better, like by actually either providing some support or actually exposing parts of it so that have nutrients coming in.
Right.
There are also engineers to create a single rosette organoid. They're more mimicking the neural tube, or actually build the neural tube-like kind of structure. They also build like a blood vessel type of kind of a path to provide nutrient. So it's actually it's a very fast growing field. And I anticipate it's going to be more and more like the brain, better model of the brain.
Right.
So the push for bettering this system is a good push. So if you put your goal there, scientists will find a way to make it happen. Right. Yeah. So and in addition to that, there's also such a thing called 3D printing. So you actually can print cells in layers or in combinations. Yeah. So my colleague Su Chun Zhang, now he actually just retired from University of Wisconsin, and so he published a paper which I actually also co-authored, is to 3D print like neurons, astrocytes, and other cell types to actually mimic the brain structure. So this is still
if somebody
very early.
Had told that 22-year-old getting off the plane in Seattle that in some number of years you'd be printing neurons with a 3D printer, you'd have laughed at
would
them.
never
No.
thought about
It is
that.
extraordinary.
Yeah. So there's like a new call from this program called Upper Edge is trying to see if you can generate human tissue and transplant it to the people with a stroke or injury. So right now, this is still kind of shooting the moon, the kind of approach, but it could be available in the future.
It has the ring of feasibility about it, right? It's clear that it's doable. It'll be challenging, but we'll get there. The science engine will get there.
Generate brain tissue and use that. Remember, you can actually take the person's blood, make stem cells, and then make those layers of brain structure and put it back to the same person.
Incredible.
And so that is possible.
Yeah.
So there may be even more impossible things we're thinking today could happen. Because actually, 10 years ago, it was unthinkable to do single-cell RNA-seq. But now we're actually doing that, right? It's almost become a regular technology, routine technology now. Yeah. So anything
who knows where
can
we're
happen.
going to be?
Yes.
And that's a very hopeful message. It really is. I always like to ask this, and maybe we'll close with this. Despite the somewhat bleak times, we can get caught up in that. At the same time, there's a very hopeful message that you just shared about just the extraordinary technology and innovation that's actually going on right now. What are you saying to your mentees, to your trainees about the scientific enterprise and staying in neuroscience and training in neuroscience? What's the message there?
So I have a lot of students and a lot of undergrads and grad student trainees. This year has been difficult because there's kind of uncertainty of funding. And some of them I actually consider now to stay in science. Yeah. And I said, science gives us hope. If you actually have any contact with parent groups and families, what they really want to hear is the hope. Even though it may take their lifetime, they may not see that happen to their own family or children. The hope actually is one of the unique aspects of the human race, right? Yeah. So we scientists give them hope. So what we do is important. So do not get distracted by current kind of stress and the uncertainty. So think about what your passion is. If your passion is to actually discover, to actually make you discover into something that you can also help, then stay on that. It is hard. It's never going to be easy. Yeah. A scientist is not an easy life, but it's exciting. It's something that actually you feel strongly, feel like passionate about it. When you reach my age, you'll be still excited to come to work every day, like what I do now. So that's what you're shooting for. So, yeah.
I think that's a fantastic, hopeful message. Thank you so much for that. Xinyu, thank you so much for being here on Neuroscience Perspectives at the University of Rochester. We're just delighted to have you. And may I add that, what a great resource you are to this great country here in the United States of America. And may it be the case that the brilliant minds of the world keep coming here to do the science the way that you've done it and in spades. So thank you so much.
Thank you. It's a great pleasure to talk to you and being here.